So, welcome all attendees and our resource person, speaker, Mr. Mihir. So, as you all know, we are organizing this free lecture series uh, under banner of Science Dialogue series. You can say this is not associated with any affiliated organization. This is only just helped by the research scholar. I have taken initiative, but several research scholar and faculties are associated with this campaign, you can say, in which today's speaker is Mr. Mihir. He did his MSc from MS University, Baroda in biochemistry department, and then he moved to Indian Institute of Science. As you all know that this is India's first rank institute, and now he is going to means get the award, PhD award. He has submitted his thesis, and you all know that it takes 6 7 years, not like in 2 3 years. So I consider that his knowledge is better than so many faculties who has completed in 2 years PhD or 3 years PhD, because getting admission in PhD is also difficult, and working there is also not its easy task. So, and I am also happy to introduce Mihir because I have taught him when I was in MS University, and I know so many story about him. So, so I called him not only to disseminate knowledge, but also his inspiration for so many students. He could not perform very good in dissertation. So, but you can see his inspiration for me also. During dissertation, his guide was not very satisfied, but he got admission in Indian Institute of Science. Means selection percentage is less than 1%. If we consider the total applicant in Indian Institute of Science, and then the final selection selected, so it's less than 1%. So, and then staying there also, you can see so many people as they cannot tolerate that pressure, so sometimes they jump from the roof, and sometimes they hang these things. So completing PhD from top institution is also a good task. You can say half you how I can say कि लाइफ का जो सबसे बड़ा जो टफ टास्क है ना पीएचडी, अगर आपने टॉप इंस्टीट्यूशन से कर लिया, बिकॉज आफ्टर पीएचडी there is very less chance to learn, है ना, under any guide. You have to learn yourself after PhD. No one will teach you, no one will guide you. So again, I'm really thankful to Mihir, and I congratulate him that after MSc, despite of not showing very good performance as per his guide things, but he secured his position in Indian Institute of Science. With this note, any students who are aspirant PhD student, you can interact after his lecture. You can interact with him. You can take his guidance for taking admission in Indian Institute of Science. And even in Indian Institute of Science, structural biology is again tough. There are four departments. One is microbiology, one is biochemistry, one is reproductive biology, and one is structural biology. So you can see there are so many CSIR directors. They are from PhD from that structural biology department of Indian Institute of Science. You might have aware of about Samir Brahmachari and so many people. They have done their PhD from that same department. So he is alumni of Indian Institute of Science. So thank you for joining us, Mihir. Now its floor is open to you. Floor is now yours for 1 hour.
Thank you, Gopal sir. Thank you for a kind introduction. You got me a little emotional there, but yeah, thank you so much. And I believe this is a good initiative that you are taking. It's important for the students to get the exposure, and I'm happy. I'm very much happy that you are providing this opportunity for them. So, good afternoon, everybody. I'll be presenting my work that I've carried out during my thesis, but I'll also introduce whatever topics that you may not be familiar with, and I'll try my best to make you understand. You can ask me any questions at any time whenever you have doubt. I think it's important because the initial slides I will explain the techniques, the basics. So if you don't understand basics, it will be difficult further on. So I urge you guys to ask questions whenever you are in doubt or have any questions, right? So I'll begin my presentation now. Can you see the full screen?
Yes, yes, it's visible.
All right. So I'll get the printer, pointer. Okay. So I'll explain basically. So there's in structural biology there are three techniques: X-ray
Click on
crystallography,
slideshow
Kravi.
mode also. Click on slideshow mode.
I think it is
नहीं
okay.
ये लास्ट ये देखो तीसरा चौथा नंबर है ना. Yes. हाँ click. Just click on that.
Show presenter view.
हाँ.
Okay. हाँ. Okay.
Okay.
Yeah. Okay.
It's not showing as it's still not showing presenter view, but it's visible to me. Any of
Okay.
you are facing any problem in seeing this slide? Because some participants are watching through mobile, then it may be difficult. Otherwise, it's good.
Okay.
It is good, but
Okay.
you have to quit like in this presentation mode in your computer. Otherwise, we won't be able to see the changes in the slide.
Okay. So I'll quit the mode, hide presenter view. Now is it fine?
नहीं यहाँ प्रेजेंटेशन मोड नीचे दिख रहा है ना, चार जो क्लिक है जो आपके हुआ.
Mihir sir,
Viewer phone, it is not visible.
which is not visible.
Okay.
हाँ बोलिए.
गोपाल सर, सर ये गूगल जो मीट है ना उसके अंदर आप प्रेजेंटेशन करें तो नहीं देगा आपको. पूरा कंप्यूटर का स्क्रीन अगर शेयर करके प्रेजेंटेशन व्यू करोगे तो आएगा.
Okay. I'll do that.
Okay. Entire
मैंने बस
स्क्रीन
presentation.
का आता है ना वो प्रेजेंटेशन में.
Okay.
Yes,
I'll
yes,
do that. I'll do that. Stop presenting this thing. And okay. So I'll say entire screen, right?
Yes.
Allow. Okay. So.
Yes.
All right.
हाँ.
So now is it fine?
Yes. और सो ऊपर जो है उसको हाइड कर देना.
Yes,
ऊपर
sir.
का लाइन. Yes.
Okay.
Yes.
All right. Okay. So I'll start by some history of structural biology of proteins. So this is the first structure of protein which was solved in 1957 by X-ray crystallography by John Kendrew and group. They also got a Nobel Prize for their work. These are the first crystals of this protein, and this protein is myoglobin. And they took almost 7 years to solve this structure, which now, if you have a good data, it is possible to solve this structure in 7 days. So it shows that how further we have come in the field of the structural biology. All the computational methods and other things have developed further, due to which we can now solve the structures within months or days. So basically, structures of proteins can be solved commonly by three methods. First is X-ray crystallography. This is the oldest method. Second is the NMR spectroscopy, and third is the cryo-EM electron microscopy, which is more recently developed. So what are the differences between these three techniques? So first of all, in sample preparation, in X-ray crystallography, you need to crystallize the sample. In cryo-EM microscopy, you need to instantly freeze your samples. And in NMR spectroscopy, you need not do crystallization or freezing. You can just solve the structure using a pure concentrated sample in solution. So this is the main advantage of NMR spectroscopy, that it gives you the structure of proteins in solution. So in case your protein is dynamic and having more than one conformation, it is important if you have this method by which you can screen, you can have an idea about the different conformation that the protein takes, which is impossible in the case of crystallography and cryo-electron microscopy, which gives you basically one photograph of your one conformation of your protein. In terms of data acquisition, in X-ray crystallography and cryo-electron microscopy, the samples are bombarded by X-rays and electrons, respectively. While in NMR spectroscopy, the sample is bombarded by radio frequency waves, which are lower in intensity, lower in frequency, which means they do not perturb the sample much through their light waves. In terms of molecular weight, in X-ray crystallography, you can study most of the molecules, however large it is. However, the crystallography is very difficult to do for the dynamic proteins, especially the peptides, and also the large molecules which are difficult to crystallize. And in that case, for large proteins, cryo-electron microscopy is a better technique, which allows you to solve the structures which have molecular weight of more than 100 kilodaltons. Nowadays, you can also solve the structures of around 70, 80 kilodaltons, as as I said before, this technique is a new technique, and it is always advancing, and they are always pushing the limits towards the lower molecular weight proteins. In NMR spectroscopy, you can study the more preferable molecular weight range is between 5 to 20 kilodaltons, and but however, in NMR also you can study they are also pushing the limit, and nowadays it's also possible to study the molecular weight of around 100 kilodalton through NMR. So, so as I said, I'll be presenting NMR. I'll be mostly focusing on NMR during this lecture, that how do we use NMR to solve the structures of proteins. So where does the NMR signal originate? Every nucleus has an intrinsic magnetic moment, which means they behave like a magnet, behave like a small magnet. However, in regular life, in general life, we don't see every object behaving like a magnet. That is because in the absence of this external magnetic field, the nuclei, every nuclei, the orientation of the magnetic field for every nuclei will be random. And because of that, the net magnetic field will be zero for any object. However, when you apply an external magnetic field, these magnetic moments distribute into two directions. The first two conformations, I would say, in one conformation they are facing the magnetic field direction, and in other conformation they are opposite of the magnetic field direction. And most of the nuclei will be facing towards the external magnetic field, which is given by this energy difference. So because of this, now the object will have a net magnetic field in the direction of the external magnetic field. However, because the external magnetic field and your resultant net magnetic field are in the same directions, it's difficult to detect, because this magnetic field, which is I have shown in as M0, is very weak compared to the external magnetic field that we apply, because of which it is very hard to detect this magnetization. So we use a trick. What do? We apply a radio frequency pulse, which basically rotates this magnetization from the axis. So now when you rotate this magnetization to 90 degree, this is called a 90-degree pulse. And when you do that, now you can detect the magnetization, because the magnetization is now in the y-axis, while your external magnetization is still on the z-axis. Another thing is that this magnetization actually precesses, this M0 is actually precesses around the magnetic field. Now, because of this precession, what will happen when you rotate this magnetization 90 degree to the y-axis, this magnetization will now also precess. And as you may have studied in your electron, in your physics class, the rotating magnetic field produces electric field, right? And that electricity is what we detect in the NMR machine. So this is a general time domain signal, and this is the frequency. This is a time domain signal, and this is the signal that we obtain. Now, what we do is we Fourier transform this time domain signal, and we obtain a frequency domain spectrum, which gives you one peak for each nuclei, right? And this is the basics of the NMR spectroscopy, that you get one signal for one nuclei. Now, this frequency is actually more generally denoted by chemical shift scale. Now, what is chemical shift scale? Is that if you have this, precession, the frequency of this every nuclei also depends upon the external magnetic field. Now, because of which, if you change this external magnetic field, if you do these experiments on 900 megahertz compared to 600 megahertz, you will get different frequency for the same nuclei. So in order to standardize that, we have developed a scale which is chemical shift scale, which standardizes this frequency So you will have a same frequency for all external magnetic fields. And this is known as a chemical shift scale. So chemical shift is also frequency, it's just a different scale for each nuclei. Now, the most important thing in NMR spectroscopy is that this chemical shift, this frequency depends upon the local electronic environment of the observed nuclei. It means that if you change the chemical environment of the nuclei, the chemical shift, the frequency will change. And this is the basic of all the NMR spectroscopy. And if because of this fact only, the field has developed into applications, into solving structure of proteins, small molecules, and all. So this actually happens because the effective magnetic field felt by the nuclei changes because of this shielding constant. And this shielding constant actually describes the chemical environment of the nuclei. So for example, this is a carbon spectrum of propanol. And these are, so propanol has 3 carbons, right? And all these 3 carbon, because they have different chemical environment, they show up at different frequencies in an NMR spectrum, right? So summarizing all this, a nucleus has a inherent magnetic moment and behaves like a small magnet. However, in the absence of magnetic field, they are randomly oriented, and because of which you do not feel the net magnetization. However, when you apply external magnetic field, they distribute into 2 states, and one state is along the direction of the magnetic field and the second is in the opposite direction. And this is because of this, there's a net difference in this populations, due to which you have a net magnetization in the direction of the external magnetic field. When you apply an RF pulse, what this RF pulse does is it moves this magnetization into the x-axis, into the transverse plane, which we call it. And because of this transverse plane, now you can detect it when this magnetic moment revolves around this z-axis, processes around the z-axis. Because the magnetic moment is rotating, you can detect this magnetization through electric in the form of the electric current. And this finally, this time domain signal is actually converted into a frequency domain, and you get a single frequency for a single nucleus. And finally, the chemical shift scale is used to denote the frequency of this NMR signal. And this chemical shift is very sensitive to the chemical environment of the nucleus, which makes this NMR very important to solve the chemical structures of the molecules. So I hope everything is clear now. If anything is not clear, you can ask me questions. All right, so I hope everything is clear. So what I will do now is I'll show you one example for which I use the NMR technique. So you'll get more idea how do we use this NMR in terms of application, right? So I'll start with the project that I did during my PhD. So this is a budget frog, also known as Lepidobactericus levis. It's found in Southern American region like Brazil and Argentina. However, because of its funny and unconventional look and intelligent behavior, it is also kept as a pet in many countries. Because the frogs are the first organisms to conquer the land, the skin acts as a first defense mechanism. And this skin secretes many types of peptides, wound-healing peptides, antimicrobial peptides, protease inhibitors, and neuropeptides, and so on, to protect itself from other outside organisms and outside environment. So one of such peptides secreted by the skin of the budget frog is called LLTIL. LLTIL stands for the Lepidobactericus levis, that's the scientific name, and TIL is the trypsin inhibitor-like peptide from family to which it belongs to, right? Now this peptide is unique because this peptide has 10 cysteine residues in an amino acid length of 55 amino acids. So it's highly concentrated in cysteines. So this is, and our lab in molecular biophysics department in IISc focuses on the study of the disulfide-rich peptides. So this is why we took up this project. Now I'll divide this talk into 4 parts. So I'll first explain the solution structure of LLTIL, that how did we arrive at the structure of this peptide. Second, I'll explain the function of this peptide. Next, I will go on to explain the relationship between the structure and function. And finally, I will also show you what are the effects of mutations on both structure and function of the peptide, right? So this is the SPS page profile of LLTIL purification. So this is the to purify LLTIL, we fused it with cytochrome B5. Cytochrome B5 is used in our lab to enhance the expression of the expression and the solubility of the proteins that we purify. And between there, we have a thrombin cleavage site. We also have a His tag, which helps in purifying the protein using nickel anti-chromatography. So this is the final purified peptide LLTL after the cleavage of this fusion protein, right? And we carried out analytical size exclusion chromatography, which gives you an idea of the molecular weight of the peptide. So what we found is that the LLTL elutes at a volume which corresponds to the 8.5 kilodalton weight protein, which means LLTL acts as a monomer. The molecular weight of this protein is 6 kDa. So we can assume that we can confirm that this peptide acts as a monomer in solution. Next, we carried out a MALDI mass spectrum, right? MALDI spectrum of LLTL. This is a MALDI spectrum of LLTL. The battery is low. I'll just connect the charger. Sorry. Okay, sorry for that. So this is the mass spectrum of the peptide, and the calculated mass was for the oxidized peptide was 633 kilodaltons, and the reduced mass was 6343. So in the reduced case, all the cysteines would be reduced. They will be in the form of SH, while in the oxidized state, all the cysteine residues will form a disulfide bond. So here I've shown a reduced form of cysteines. When this disulfide bond forms, both the cysteine oxidizes. So per 2 cysteine residues, you'll have a proton will leave, 2 protons will leave. So for each cysteine, 1 Dalton difference will be there, and because of which the oxidized mass would be 633 Dalton. And this is what we observed also in our mass spec data, which means that all the cysteine residues in the peptide are in the oxidized form, which means that all the cysteine residues are forming disulfide bonds with each other. So there are 5 disulfide bonds in this peptide. Okay.
Hello. क्या हुआ?
Spectrum. Each proton in the
इंटरनेट
protein.
चला गया था, लग रहा है, हैंग हो गए थे. Okay.
Oh, I see.
Just 30 seconds के लिए. Yes, continue please.
Yes, it is
Okay.
okay.
This slide from fast.
Sorry.
Please repeat this slide from fast because
Yeah, sure.
here was not
Sure. Okay, I'll do that. So this is the proton NMR spectrum, 1D proton NMR spectrum of LLTIL. So in this spectrum, each proton of the peptide is represented. So that's how sensitive the NMR spectroscopy is, right? And this is what we call an amide region of a peptide. So this is where all the amide peaks of amide protons of the peptide will appear. Now each amino acid residues have only one amide backbone amide group, and because of which this amide region actually represents each residue in the peptide. However, you can see that the many signals are actually overlapped, and it's hard to differentiate which signal comes from which peptide, right? It's because the distribution of these peaks is distributed between 6 to 10 ppm. And the sensitivity is not enough to differentiate between these signals. However, what information we can get from this spectrum is that information about whether the peptide is folded or not. So if the peptide is in a linear form or a disordered, what will happen is that it will experience same chemical environment because each amino acid is exposed to water, and they don't have the complicated if you don't have a complicated 3-dimensional structure, a globular structure, you won't have difference in the chemical environment for each amino acid. And because of that, in the case of disorder proteins, your peaks will be very distributed, very narrow in the chemical shift scale. So if, for example, if you see here, this is a disorder protein called VPG. This is just a pictorial representation. And you can see that the peaks are distributed from 7 to 9 ppm. However, here the peaks are distributed from 6 to 10 ppm. Now, this is because the peptide is well folded, because of which each amino acid experiences different chemical environment. Because if you have a tryptophan, if it's surrounded by, let's say, lysine, glycine, and some other amino acid, there will be a unique chemical environment. If it's disordered, then the tryptophan is only surrounded by the water and the nearby residues. So because of which you see this dispersion in the chemical shift scale, which is a good indication whether the peptide is folded or not. So we can say confidently that in this case, the peptide is well folded. Next, to solve this problem, to assign this each peak to each amide proton in the peptide, one has to go further and do 2D heteronuclear NMR spectroscopy. So to do 2D heteronuclear means you'd use not one nucleus, right? You don't use only proton, you will also use carbon, you also use nitrogen. However, the generally commonly found isotopes of carbon and nitrogen, C12 and 14N, are not NMR active. The NMR active isotopes are 13C and 15N. So what we do is we grow the cells in the presence of 15N ammonium chloride and 13C glucose in minimal media, which means now the cells have the source which is only N15 and 13C labeled, due to which your peptide gets labeled by 15N and 13C isotopes, right? Now this is 15N HSQC spectrum. This is a 2D NMR spectrum. So what we see here is that for each proton, so for example, the peak here that you see corresponds to this peak, right? Now each peak, each of these proton chemical shifts is also correlated with your nitrogen chemical shift, right? So this is a nitrogen chemical shift to which it is attached to. So this is an amide pair. So this is a peak for the amide pair of the single residue, right? So you see this amide pair for each 55 residues in this spectrum, except for the prolines, because prolines do not have the amide proton, right? So except prolines, each amino acids appear in this spectrum as a peak. And as you can see, compared to this 1D spectrum, all the peaks are very well differentiated. So now we can perhaps go ahead and assign each peak to the single amino acid residue in the peptide sequence. So that is our next goal. Now to achieve next goal, what we do is we go further and we do 3D experiments. So in 3D experiments, you don't use 2 nucleus, you use 3 nucleus. So in this case, for example, we do 2 experiments. These are 2 complementary experiments where we observe the proton, nitrogen, and the carbons. Now the difference between these 2 experiments is that in CbCSU-NH experiment, the amide pair is correlated with the carbons of only previous residue. While in HNCACB experiment, the amide pair is correlated with the previous residue carbons as well as same residue carbons. Now, using this information, what we can do is we can connect the residues which were seen in the HSQC spectrum. So this is what it is that this is the 3D strip of only a single peak that you have seen in HSQC. So whatever you see in here, this peak here is extended in the 3 dimension, in the 3rd dimension. And this is the 3rd dimension which shows the carbon chemical shifts of the amide pair. So this each strip corresponds to one single amide pair, and this each amide pair, because it belongs to a single residue, the each strip we can say belongs to a single residue. So now we have the connectivity between the residues. So we know that this residue is connected to this and this and this and so on. However, we still don't have the information that which strip corresponds to which residue. We only have connectivity, we don't have the assignment. To obtain the assignments, what we use is the unique chemical shifts of the C alpha and C beta carbons. Now, if you observe here, the C alpha carbon of glycine has a unique chemical shift of 45 ppm. No other C alpha of any other amino acid comes at 45 ppm. And here you can see the C alpha comes from 45 ppm. So we can assign this residue as glycine, right? However, in our sequence, there are 5 glycines actually. So we don't know which glycine this is, right? Now, if you notice here, the first glycine, the second glycine, and the 4th glycine has serine or threonine after it, right? And if you look at here, the serine and threonine, the C beta chemical shifts of serine and threonine are unique. They are around 65 to 70 ppm. So what we have to see the C beta chemical shifts of this residue here, and this is actually at 35 ppm, which comes around here. So we can rule out these 3 glycines, right? Now what remains is the 3rd glycine and the 5th glycine. So I'll show it here. So what remains is the 3rd glycine and the 5th glycine residue. And if you have this 3rd glycine residue, then the 7th residue will again be glycine, right? So from here, the 7th residue, we should expect glycine, which is not there because there is no 45 ppm chemical shifts of C alpha carbon, which means we can rule out this glycine also, which means the glycine that is left is only this glycine, and we can assign this sequence as the sequence present here. So basically what I wanted to what message I wanted to send you is that this NMR spectroscopy is like a puzzle. So it's very interesting if you are into puzzles and if you like to solve puzzles, this technique will be very interesting because in X-ray crystallography and cryo-EM microscopy, all you get is a picture, right? But NMR spectroscopy is an indirect method. So you have to use many kind of ingenuity, right? And you need to do many kind of experiments to figure out the sequential assignment for these residues, right? So this is a final assigned N15 HSQC spectrum of LLTIL. And here you can see each peak is now assigned to a residue in the spectrum. This spectrum is called the fingerprint spectrum of any protein, any peptide. And this is a starting point in the structure determination by NMR. And the advantage of this, even if you don't want to solve the structure of a protein, what you can do with this spectrum is you can study the interaction with other proteins, nucleic acids, small molecules, and cofactors. You can do this because, as I told you before, the NMR spectroscopy is very sensitive. The chemical shift here is very sensitive to the chemical environment of the protein. If you add the other protein or any other external agent, if it binds to the protein, if it binds to some residue, some selective residues, then only the chemical shifts of those residues will change because of the change in the chemical environment. So you can track the changes in the position of chemical shifts and thereby predict which residues are important in the interaction with other proteins and nucleic acid, drugs, and other small molecules, right? Now I will explain how do we go from this to the structure of the peptide, right? Now to do that, we also need to assign other nuclei. So using different experiments like CCCONH, HCCONH, I won't go into the detail, but we were able to assign almost all the nuclei present in the peptide. Now, once you have this information, you need these 4 kind of major information to solve the structure of the peptide or protein by NMR. You need the information on the dihedral angles. You need the interproton distances. You need the information on the disulfide connectivity because our peptide has 10 cysteine residues. You need to know which cysteine is connected to which cysteine. You also need the information on hydrogen bonds. So in next 4 to 5 slides, as I explain how we obtain these 4 information using NMR spectroscopy, right? So coming to the backbone dihedral angles, this is the phi and psi angles. These are the backbone dihedral angles.
Sanjay Bharadwaj has some question. Please unmute and
Sure.
speak, Sanjay ji. Sanjay Bharadwaj.
Hello, am I audible, sir?
Yes, you are
Yes,
audible.
yes.
Please ask.
Sir, actually, even I work on NMR spectroscopy, but just small molecules. So I want to know how is the sample preparation for this protein, like for proteins when you analyze? It's just you're using D2O or like what exactly?
So for sample preparation, we purify the peptide without D2O. We only purify it in water in buffers. And after we get a concentrated sample of, let's say, so we generally use around 300 micromolar to 500 micromolar for this triply labeled samples. And when we purify, when we get the concentrated sample for, let's say, around 1 ml or something like that, what we do is we add 10% D2O externally to lock the sample in NMR spectrometer. So we use only 10% D2O in our final sample. I hope that was your question, right?
Yes, sir. Yes, sir. Even I've used 10% T2O for some biological samples. And
Yes, yes.
which internet standard do you use, sir? DSS or anything else, sir?
We use DSS.
Okay, sir. Okay.
Use
Sir,
DSS.
and another doubt is you finally immediately you assigned all the carbons and you showed the 2D HSQC, right, sir? Like where all the
Yes.
carbons are assigned. So how
This-
you exactly, because many carbons in the previous slide was having overlapping, like the region of the chemical shift is almost same, like of many residues. So how could you do this slide, sir? Like how could you assign each carbon?
So what we do is we do not. So what I have shown is only 2 experiments, right? For this one, we have only shown 2 experiments, but we also do further experiments. So I'll show you. For example, here we have the information on C alpha and C beta, right? But we also do other experiment. For example, CCCONH, which also gives you the same information, but it also gives you extra information about the other side chain carbons also. So what we do is, in case of an overlap, we try to make sense of it by using different experiments, right? So we may increase the dimension, or in the case of, for example, if you have HNCACB spectrum, there you will have more overlap compared to the experiment which is CBCACONH. Because in HNCACB spectrum, you get the information of the carbons which are both in the current residue and previous residue. While in the other experiment, you only get information about the previous residue. So in case of the overlap, we always try to simplify the spectra by choosing the experiment which will give us the less number of peaks. Right? So that's what we do. And you are right. In fact, in some cases, even after doing that, we are not able to resolve the overlap. So in that case, we just guess. We just go by our gut instinct, what we feel is right. So this and
Hello.
Yes, sir. Yes, sir.
आवाज आ रही है उनकी?
No, sir.
इंटरनेट का देख.
Hello.
Yes, yes. चले गए थे. इंटरनेट चला गया था.
Oh, okay, okay.
लेकिन बता दिया था. ज़्यादा नहीं था, है ना?
Okay. Fine. So is it fine? Was I able to answer the question?
Yes, sir. Yes, sir.
Okay.
And, another thing is like, sir, what exactly the parameters that are
So let
important?
Sanjay ji, please let him finish then, because you have so many
Yes.
questions. So one
Yes,
to
sir.
one after finishing, you can, है ना?
Sure, sir. Sure,
So many
sir.
participants are waiting, so they will learn, and then you can talk.
Yes, sir. Yes.
Yes, it's good. The interaction is good, but let him complete, है ना?
Sure, sir.
I will come back to you, right? I'll come back to you in the end. Okay. So, okay, fine. So the first information is about the backbone dihedral angles, which are phi and psi. So this dihedral angles were predicted by the empirical method. So what we use is, there is a tripeptide database which is available, where you can you have chemical shifts. So this is always an empirical thing. So you have the empirically derived dihedral angles from chemical shifts, and using this, so these are all present in the Talos Plus facility. And using this facility, what we do is we predict the dihedral angles of phi and psi. So after predicting this, one can get information about the secondary structure of the peptide, right? And what we found is that this peptide has very limited secondary structure. It only has 4 beta strands, and these beta strands are also very short, right? And most of the peptide is actually contains loops, right? And it's disordered. It's not completely dominated by secondary structure, as you see for many proteins, right? So this is a interesting thing. Next, we can also determine the chi1 angle. Chi1 angle is the dihedral angle between the C alpha and C beta. And this angle can exist in three forms: Gauss plus, Gauss minus, and trans. So using the experiments HNHB and HNCOHB, one can actually determine which angle the residue takes, right? In HNHB, the amide pair is correlated with the beta proton of the present residue, current residue, and in HNCOHB, the amide pair is correlated with the beta proton of the previous residue. And using the from the relative intensities of these two beta protons, one can actually determine. So for example, if you have, so in the case of 1652, one beta proton is in high intensity compared to the other one in the HNHB spectrum. In HNCOHB spectrum for 1652, both the protons have very low intensity, which corresponds to this situation here, which means one can assign this 1652 chi1 angle to minus 60, right? So likewise, we have determined the chi1 angle of totally 27 residues from this experiment. Next, the most important thing is the atomic distances, which is important to solve the structure. And these atomic distances are actually extracted from the effect called nuclear over-Heisler effect. So nuclear over-Heisler effect is actually gives you an idea about the atomic distance between the two nuclei. And the intensity of this signal is proportional to the distance in a proportional to the distance by 1 upon r6 r is to 6, right? And these NOEs are actually can be sequential, medium range, or long range NOEs. So basically, what this NOE tells you is that if right, so pointer use laser pointer. Okay, so as we seen in the initial slides, the oxidation state of cysteines was determined using mass spectrometry. And NMR also can provide you information about the oxidation state of cysteines. For oxidized cysteines, the C beta chemical shifts will be more than 35 ppm, and that is what we observe here. So we can confirm the mass spec data that all the cysteines are oxidized, forming 5 disulfide bonds. The disulfide connectivity was determined through the NOEs, right? So if the two cysteines are in proximity, we assume that they are disulfide bonded. So we found NOEs between these 4 pairs, but we could not find NOEs between these pairs because of the signal overlap. However, we know that all the cysteines are oxidized and disulfide bonded, so we can assign this pair also as a disulfide bonded pair. So likewise, we could assign this disulfide connectivity of the peptide, and this is the final disulfide connectivity of the peptide. Next, there is also something isomerization called as proline cis trans isomerization. So prolines can exist in cis or trans conformation, and if you have this C beta, C alpha, C gamma chemical shift difference of around 10 ppm, then we can assign them as cis conformation. And if we have this chemical shift range between 4 to 6 ppm, then we can assign them as in trans conformation. And that is what we find for all the prolines in our peptide. We have 5 prolines, and for all 5 prolines, we find that they have chemical shift difference between 4 to 6 ppm, which means all prolines are in trans conformation. Now coming to hydrogen bonds are very important in solving the structures of the proteins and peptides. So this is a very good, very direct experiment to detect the hydrogen bond. So in this experiment, this is called a long-range HNCO experiment. So in this experiment, what we do is we correlate the amide proton to the carbonyl carbon of the hydrogen bonded residue. So for example, here the amide proton of the lysine 10 is correlated with the carbonyl carbon of the phenylalanine 39, which means they are disulfide bonded as shown here, right? So this is how we were able to get direct information about these hydrogen bonds using this long-range HNCO experiments. And we found these hydrogen bonds across this beta 1 and beta 2 sheets and beta 3 and beta 4 strands, not sheets. And accordingly, we can say that this forms the first beta sheet, beta 1 and beta 2 strands, and beta 3 and beta 4 strand forms the second beta sheet in our peptide. And in total, we were able to detect 17 hydrogen bonds. So now, equipped with all this information that we obtained from NMR spectroscopy, we moved ahead to solve the structure using Cyana. So in Cyana, what we do is we have all this information about dihedral angles, hydrogen bond, disulfide connectivity, distances. We make it into a single file and we feed it into the software. And the software, what it does, it calculates using some algorithm, it calculates the structure, and it shows you and it gives you the structure, right? And there will be some violations. So so as I told you before, right, NMR is not an exact technique, and there may be an error during the assignments, right? And if there is an error, it will show a violation. So what you do is, if it shows the violation, you go back to your distances, your spectra, and you correct your assignment and you do the calculation again. So after doing many cycles, ultimately you have to come to a point where there is no violation, right? And once you have the structure, you can move ahead with the structure refinement also using water. So what you do is you put your peptide in a water computationally, and then you refine your structure using water refinement. And then also you will get some violation. Again, you go back, correct your assignments. Again, you come back. So this is how the whole cycle goes. And most commonly for these small proteins, this structure calculation can take up to 2 to 3 months. So NMR is comparatively takes a longer time compared to the other techniques like crystallography or cryo-electron microscopy.
How many slides left?
So I think there are many slides left. How much time do I have? I only will a little time. Sorry?
1 घंटे का ही दिए थे, means you have more 7-8 minutes.
Yeah. So what I'll do is I'll only explain the structure, and then I will explain the ITC one time, right? So I'll do it. I'll finish in 3 to 4 slides.
In complexity of the structure, है ना?
Sure.
मुझे भी तो 10-15-20% ही समझ में आया. मुझे भी
No problem. That is much more. So, okay, these are the final ensemble. So in NMR, we don't get a single structure, we get multiple structures. So these are final ensemble of 20 structures that we get from NMR spectroscopy. And these are the list of constraints that we use to solve the structure. And this is the final structure statistics, which shows that RMSD between these structures is 0.76 angstrom, which is considered good. Right, and this is a Ramachandran plot that you must be aware with, and this Ramachandran plot. So every residue in this peptide comes under the favored or additionally allowed region of the Ramachandran plot. So the Ramachandran plot is very much satisfied in this case, which means that these structures are of a good stereochemical quality. Right. This is the final one structure of the lowest energy structure of the peptide. These are the disulfide bonds. These blue sheets are the blue strands are the beta strands. And as I said before, this structure lacks the secondary structure, right? And there are many loopy regions. However, these loopy regions are actually stabilized by these disulfide bonds. If you observe here, the loop 2 has highest length of the loop. However, there are 3 disulfide bonds present in this loop, which connects this entire loop to the structured region of the protein. So now I'll just explain. So I won't go into the detail. What we ultimately found was that this peptide is actually an inhibitor of subtilisin. So subtilisin is a microbial protease. And using many functional, enzymology studies, we found that this peptide inhibits subtilisin, a microbial protease, thereby protecting itself from other microorganisms. So next, what we did is that after characterizing the function, we wanted to find out the structure-function relationship. So what we did is we did 3 things. We did analytical size estimating chromatography, NMR, and ITC. So in analytical size estimating chromatography, you see that when you add this both LLTIL and subtilisin, which it inhibits, when you add both of them together, it forms a complex in the size estimating chromatography, which is also observed by STS page chromatography. This is the STS page of this peak, which is a complex peak. And as you can see, the both subtilisin and LLTL present, which means LLTL, my peptide, and the subtilisin, which is a microbial protease, they form a complex. Now, to get an idea about the energy parameters for this interaction, we use ITC. So ITC is isothermal calorimetry, and it gives you an idea about the thermodynamic parameters for the protein-protein interactions. Both these LLTL and subtilisin are proteins. So in this case, what we have is a reference cell and a sample cell where we have the sample. And in reference cell, we only have the buffer in which a protein is actually solubilized. And in the syringe, we have the titrant. So in this case, what I had was I had a subtilisin in a sample cell, and then I had LLTL in my syringe. So when you what we do is I added LLTL from the syringe, and as I add LLTL, there is a change in the heat, which is maintained constant by this power supply. So the dip in this power supply actually indicates the binding. So this is the experimental data which shows the binding of LLTL to subtilisin, and when we integrate it, we get the heat exchange, the enthalpy of binding here, and this is the molar ratio. This is like as we increase the concentration of LLTL, we find that only around 1:1 ratio, the binding is almost saturated here, and the binding constant comes down to 31 nanomolar, which corresponds to very tight binding. If you have a tight binding, you'll get the dissociation constants in nanomolar or picomolar range. And the enthalpy change here is -11, and as you can see here, kilocalorie per mole is -11. And you have the delta G, and you can also get information about delta S. So you get all the thermodynamic parameters plus the stoichiometry. This N is the stoichiometry, which is also 1. So this is what you get from ITC. And I think my time is up here, so I'll just stop here.
खत्म हो गया ITC थोड़ा सा और ब्रीफ में बता दो नहीं तो आप ITC का.
So in ITC, what we can do is, so if you understood this, I think the basics are more or less covered, right? If you have, so what it does is that, I don't know where was I disconnected, but this instrument here, what it does, it maintains the temperature. So this is the basic premise of the ITC, that if you have the reference cell where you have the, where you only have the buffer, and you have the sample cell where you are injecting a titrant. So in this case, I'm injecting LLTL. So what happens if the both protein molecules bind, because of the binding, there will be a difference in the binding will be either endothermic or exothermic. So there will be a change in the temperature of the sample cell, right? And because this instrument actually maintains the temperature of both the reference and sample cell, there will be a power supply change of the sample cell to maintain the temperature. And this power supply is actually what we observe here in this spectrum, if you see here. And using this, we convert it to heat. So that is what we do. We convert the power to heat. And this is the basic premise of ITC. So you can actually characterize many binding events through ITC: protein, nucleic acid, protein, small molecules. So in this case, both are the protein molecules. In some other cases, if you have protein and a small molecule, generally what we do is we put the protein in the sample cell and we put the drug or small molecule in the syringe, and you can get the profile of this whole thermodynamic parameters. And if I talk experimentally, it's a very easy experiment. It can be done within a day if everything is standardized and working well. But that is rarely case in our.
So what is the cost of this instrument?
Sorry.
Cost क्या होगा इसका क्या है?
Machine का cost. I think it will be. I'm not sure. I'm not sure actually, but it is quite costly. That I know.
Okay, because there are so many alternate methods available for protein interaction.
Yes,
so
definitely.
Only protein interaction, na?
Mm-hmm.
This method. So there are pull down.
The advantage of this technique is it's very non-invasive. So you can also do this experiment using, if you have a fluorescently labeled marker, right? I think that is one way to do it. But that is very invasive because if you allow it, if you tag it with other molecule, the inherent binding can be affected. So this is just like NMR, right? ITC is also very non-invasive, right? It does not affect the binding event. So that is why I think ITC is more preferable compared to the all the fluorescent based
Not
techniques.
fluorescent like co-immunoprecipitation. So if we have antibody against one protein, we can
Right.
do this pull down type of experiment.
Yeah, that you can do. That you can do. But that again, I guess you have to bind it to the antibody and all those things, right?
Yes.
You have to do that. So that is avoided here. You have only two moiety.
So
I guess that is.
yes, biophysical techniques are difficult, and at least for biologists, it's difficult. But you have tried to make us understand. That's good. Any question from any participants, any attendees? Sanjay ji, now you can ask. Before Sanjay ji, because he's doing experiment on this thing, so he may have so many questions. So tell me, that 6 kilodalton protein was observed under SDS page, or you have made some changes? What you have done?
So yes, we purified it and we observed.
Because generally less than 10 kilodalton protein is we use trypsin SDS, which can give better resolution. And your institute have all this facility, because here
Yes.
you have last band of 10 kilodalton, so it's merging. You
Yes.
cannot calculate, and so whether it is 7 kilodalton or 8 or 9 or 10,
Correct.
everything is there at the bottom.
Correct. So what we are interested actually in this STS page profile is not the molecular weight, but we just want to see whether, because this is a fusion protein, right? This is with the whole thing. This is the whole thing. And when we cleave it, we get these two parts. So we cleave it using this thrombin cleavage, and we get this is the cytochrome and this is VPG. This is the LLTL. So these are the two proteins.
So which vector you have used for producing this recombinant?
This is, I think, in PET21.
PET21 have this thrombin proteasoids?
No. This whole construct is actually included between the MCS.
Okay.
Yeah.
So now any question from any participants? कोई question नहीं है? So you don't forget to write your feedback in Google form, so that it will help us to improve. It will help a speaker to improve. So there is always a scope for improvement. That's why we take your honest feedback. So write positive thing, write negative thing. Sanjay ji, are you here? Sanjay ji left. Sanjay
Yes,
Bharat, yes. Now you can ask question, so that within 5 minutes we will conclude. So that next lecture will start at 4 p.m. on virus.
Yes, sir. Sir, I wanted to know what are the NMR parameters that we should know before setting up this 2D experiments for protein, like any important parameters like mixing time, all those things.
Yes, sure. So I guess for smaller molecules, what you do is you use long delays, right? The D1 is generally in seconds, around 5 seconds or 10 seconds you take. While in case of proteins, I guess what we do is we use a smaller, like 1.5 seconds is enough, because the relaxation is faster for the bigger molecules, right? So that is a major difference that you have to take into account. And of course, the pulse widths you have to calibrate for all the nitrogen and proton. And for other things, I guess for 2D and 3D experiments, you have to make sure the number of scans in the indirect dimensions, number of points in the indirect dimension. So for example, in the case of nitrogen HSQC, you have to make sure that what will be your spectral width, what will be your number of points, because that will determine the resolution in the indirect dimension. So I guess just like in 1D you don't have to worry about it, right? Because you always get, you can take as many points as possible. There's no time limit. But if you do a 2D experiment, if you increase the number of points in the indirect dimension, so the time of the experiment will increase. So you have to make sure how will you get optimum result from your experiments using as little points as possible, because that will increase the experimental time. So that is the most important thing in 2D and 3D and other experiments, I guess, to make sure you allow number of points which are sufficient to give you good resolution, and also make sure that it does not take too much time that your protein will degrade during the experiment.
Sanjay ji, can you introduce yourself? Hello. संजय भारद्वाज जी.
Hello.
Yes, yes,
Hello. Yes, sir. Am I audible?
yes, yes,
Yes,
you are
yes.
audible. कैमरा भी ऑन कर
Were you
लेना.
disconnected?
हाँ, बोलिए.
I work in CFTRI, sir, as project associate, working on like food samples like carbohydrates and not proteins, only carbohydrates. So I wanted to know like how they do on proteins.
Okay, very good, very good. So keep in touch, because today only one company people they have asked me if there is some people who can coat the vegetable vegetable coating का कोई expertise हो. It's good. And now, so a student, if you are pursuing PhD or you wish to know about the anything about Indian Institute of Science, you may ask question, or you can also share feedback. Vitra Bhattacharya जी, सुशांत,
Yes, sir.
आपका
Yes.
PhD चल रहा है?
Sir, मेरा?
हाँ,
No, sir. I'm BSc student.
BSc तो आपको ISC में इंटीग्रेटेड PhD के लिए आप पूछ सकते हो वहां पर. मैं कल्पना
Okay, sir.
means BSc MSc student can know about environment, working environment, how to get admission, all these things, or in future
Oh,
also. So
sir.
you can join me and I will connect you to the Mihir. You all, if you have not joined on LinkedIn, you can join on LinkedIn also.
I
Acha.
think someone has asked a question.
Yes,
Said
yes.
about summer internship. So
Yes.
yes, they are available. In fact, if you what you have to do, you have to mail the guide that you want to work with. So in MBO only, many students from MS University has come to do their internship, right? Especially from the integrated course we have, right? So in Gopalji, we have it in MSU. So from that department, many people come to do the internship. So all you have to do is you have to, if you have an interest in particular lab, you can write it to the PI of that lab. And if there is a space available, if there is some work that can be done, they will surely invite you.
समझ में आ
So
रहा
that
है.
you can.
समर ट्रेनिंग के लिए आपको उस गाइड को अप्रोच करना पड़ेगा, एंड यू हैव टू जस्टिफाई व्हाई यू विश टू वर्क अंडर हिम और हर. उनका काम समझना पड़ेगा. Otherwise, Indian National Science Academy invites application for summer training. You can apply there also. But किसी भी गाइड को आपको कन्विंस करना पड़ेगा कि उनके लैब में how you will contribute. So you have to understand his or her project टॉपिक. ऐसा नहीं कि हम करना चाह रहे हैं, सिर्फ ट्रेनिंग सीखना चाह रहे हैं. नहीं, आपको उसमें समझ के बताना पड़ेगा कि आप वहां जाएंगे तो क्या करेंगे. देन वो आपको लेंगे कि चलो भाई. नहीं तो बहुत सारे ऐसे लोग हैं जो एमएससी बीएससी में सब इंडियन इंस्टीट्यूट ऑफ साइंस, आईसीजेबी, जेएनयू, आईआईटी जाना चाह रहे हैं. आपको जस्टिफाई करना पड़ेगा कि आप उस एमबीयू लैब में जाना चाह रहे हैं, कुछ स्ट्रक्चरल बायोलॉजी लैब में जाना चाह रहे हैं, माइक्रोबायोलॉजी. तो वहां आप उनके किस चीज से इंटरेस्टेड हैं, क्यों आप उसी लैब को चूज किया. यू हैव टू जस्टिफाई दैट, व्हाई यू हैव चूज दैट लैब, व्हाई यू विश टू वर्क देयर इफ यू विल गेट अपॉर्चुनिटी. दिस इज कॉमन ट्रेंड एवरीवेयर. पोस्ट डॉक में भी लोग पीएचडी के बाद भी अप्लाई करते हैं. तो यू हैव टू जस्टिफाई व्हाई यू हैव चूज दैट गाइड सो दैट यू कैन वर्क देयर एज ए पोस्ट डॉक फेलो. है ना.
Yes, yes.
आपको यही क्राइटेरिया, कोई एग्जाम नहीं होता है जहां, यही क्राइटेरिया है साइंस में कि आपको कहना पड़ेगा कि भाई वहां जाएंगे तुम क्या रिसर्च करेंगे. है ना.
Yes,
आप
yes.
वहां क्या वैल्यू.
काफी लोग अप्रोच ही नहीं करते हैं. सो आई वुड विश कि ये लोग थोड़ा और अप्रोच करेंगे. सो इट्स नॉट दैट डिफिकल्ट. If you approach them with the right intention, they may give you. So yeah, statement of purpose. Somebody has said statement of purpose.
SOP तो
वो जरूरत नहीं है. So you don't have to write what you want to do, because generally if you are doing internship, you don't have to get a project with you. So what you basically need to do is you just have to show interest, that interest in a technique or in the method, right? So you can say that I'm interested in learning how to purify protein. I'm interested in learning how NMR works. I'm interested in learning how to do crystallography, right? So only that you have to write, because projects they will have, and only then they will hire you.
So I will connect him. I will tag him on LinkedIn post when I will write. So you can connect with him. Rasmi, है ना. नंबर भी दे दूंगा, आप ले लेना नंबर, बट लिंक्डइन पर ज्वाइन करो. दैट विल बी बेटर. है ना.
Yes.
Yes, sir. Yes, sir.
Sure.
Thank you.
Yes, sir. So thank you Mihir again for joining.
Sure. I hope कि helpful रहा होगा सबके लिए. And
बिल्कुल थोड़ा सा इनिशिएट होना जरूरी है, क्योंकि हम लोग
Yes.
बायोलॉजी वाले बहुत सारे जो फिजिक्स का मैटर है, मैथमेटिक्स का है, उसको टच ही नहीं करते हैं. सो दिस पार्ट्स आर अनटच्ड. तो इसके लिए इनिशिएट तो करना पड़ेगा ना. मैं तो सिर्फ मोलरिटी नॉर्मलिटी पढ़ाता हूँ बीएससी एमएससी में, है ना. वो भी बहुत डिफिकल्ट लगता है बहुत सारे बच्चे को. और ये तो एनएमआर तो हमको भी डिफिकल्ट लग रहा है, हमको भी कुछ समझ नहीं आ जाता. बट जब समझना चाहेंगे 2 बार 4 बार तब तो समझ पाएंगे ना. बिल्कुल छोड़ देंगे
Sure.
तो हम पूछते हैं एमएससी में बच्चे को 0.2 into 0.2 कितना, तो बोलेगा 0.4, है ना.
I think Faiza has said something. Thank you, Faiza, and I hope you understood it. And
जाई जाओ
Anamar?
फैकल्टी.
Yeah. Oh, okay.
है ना.
Okay. I see.
These
Yeah, I hope.
also.
Okay.
Okay. So thank you very much all the attendees and our speaker, our student Mihir. It's good. All the best, Mihir. All the best.
Thank you.
Now you all are welcome to join at 4 p.m. lecture on virus. He will take lecture on virus, and he is at Gujarat Technology University. And so you all can join. Thank you.
Okay. Thank you.
Thank you, sir.
Bye.